my great pleasure to introduce Anna Duggan. Anna is the Associate Chief, or Acting Chief, sorry, Computational and Operational Genomics at the National Microbiology Laboratory. She's also an adjunct assistant professor here at McMaster University. And she did her PhD at the Max Planck Institute for Evolutionary Anthropology. So she has quite the specialty. And then she came to McMaster after that for a few years, where she was postdoc in the Poynter Lab. So go ahead, take it away, Anna.
Thanks. Yeah, my pleasure to be here. And as I mentioned, if there's questions at any point, please feel free to just interrupt me. I don't mind that at all. Yeah, so saying that I have a specialty is probably true, but I think that I came to it via a very meandering path. It's not that I had a single focus from the very beginning and stuck with it. I have changed my focus several times, I think, which I think is, there's an argument that it can be a detriment in that your publications are kind of all over the place. But I think in terms of skills, it's actually a really beneficial thing in that you try out a whole lot of different things and you find the one that you really like and enjoy and hopefully learn some lessons from the other ones as well. So in terms of using a PhD, I think first you have to get one. And also not a terribly straight path, even though it was kind of straight through education. I did my undergraduate degrees at the local university, Memorial University of Newfoundland, sometimes now just called Memorial, but I will insist until the day I die of tacking on of Newfoundland. Yeah, the local university. I grew up, 10 minutes outside St. John's, and curiously enough, I don't think there was ever a question that I wouldn't go to university. And so I was actually in undergraduate degree before I thought, do I actually want to be here? Is this what I want to do? It seemed like the path that was destined for me. Both of my parents had gone to university. Both my parents had gone to MUN. They were both the first generation in their families to go to university. So that plus, the early 2000s heyday of you must have a degree to have a successful job, just made it seem like it was the only path. Even if I wasn't necessarily terribly happy about it, I really wanted to go away for school. I really wanted to go to Dalhousie because Nova Scotia felt, exotic and far away. But ultimately, I think I got a really good undergraduate education at MUN, even if, there's a little bit of inferiority complex associated with it. It was then, and I think it still is, the largest undergraduate university in Atlantic Canada. Some of the infrastructure was not great, is not great, but the quality of the education was really, really high. And I didn't appreciate that until after I had left it. So I was there for 5 years. Part of that was that I didn't know what I wanted to do. As I said, it seemed like it was the place that I had to go, and I didn't think about why I was there until I was there. Didn't have the greatest first year. Took the courses that I had to take, didn't always do great in them. I was never going to have a 4.0 GPA to get into med school, but I also didn't really want to go to med school, so that was all right. It took me about a year to realize that I was probably a biologist who had never taken a biology class, mostly because I had really disliked plant biology in high school. So I had these blinders about what biology was. And when I finally got in there, I got really excited about genetics. And being excited about genetics might sound silly in 2023, but in 2004, 2005, in the biology department at MUN, that's an important caveat there. It was a kind of novel thing. The department at that time was very, very focused on fisheries, ichthyology, ocean sciences, so much so that they've actually created their own ocean sciences department now. But at that point in time, the biology department was essentially an ocean sciences department. So I focused as much as I could on molecular biology. That meant mostly I was doing wildlife genetics, so looking at mitochondrial DNA for population structure of codfish, caribou, other native species to the island, essentially, using the, at that time, cutting edge, but very quickly, very obsolete technology of DNA sequencing microarrays. They were a flash in the pan, but I was there for that flash. I was there for 5 years because I also did a BA at the same time. I did a bachelor's degree in Russian language and literature, which is something that I routinely forget about until I look at my CV or I do a talk like this. And the reasons for doing that were not academically strong. They were more so social reasons, social and experiential. But I think it gave me, again, a different set of skill sets. It was, switching your brain between one set of classes and the other and really getting to exercise different ways of thinking. And ultimately, I think it was really beneficial. So I then went to Queen's to do a master's degree. It was again in biology. I wanted to focus still on questions of molecular biology and evolution, but less so genetics. So I wound up doing a project that was kind of about protein evolution. It didn't work. The Queen's years for me were really about surviving, not thriving. And part of that was just that my projects kept failing over and over again. So antibodies that turned out to be nonspecific, then, spending months growing up cohorts of fish in different conditions to try and compare what their gene expression looked like in different temperatures, only to have the fish all get sick and die. It got to the point where I had a conversation with my thesis supervisor, and I said, I just want to finish. I want to get out of here. I'm ready to be done. So I have a master's degree from Queen's University. I have to look up what my thesis is called anytime I need to reference it. And once I had a conversation with a former lab mate about some follow-up work that he had been doing, and he said, well, yeah, these new findings really go against things that have been previously published, and he explained it all to me. And I said, oh, that's so interesting. Who published that first study that yours are all disproving? And he kind of blinked slowly at me and said, that was your thesis, Anna. It was an experience. I did it. I got the piece of paper. The 2 years at Queen's are the reasons why I've told many people over the years that you can do anything for 2 years, because I did that and I got out the other end, but I was really ready to be done and move on. Not a glowing recommendation, but it had very little to do about the university itself or my supervisor, who was a lovely person. It just, it was not a great combination of factors in those particular years. So then I decided I needed a really big change. And I realized that what I'd always been most attracted to in molecular biology and genetics was fundamentally about people. So understanding not so much what makes different groups of people distinct or different, which is how I think a lot of human genetics gets interpreted or focused, but instead about what really binds us all together. So how do we share this, phenomenal human history over the last 200,000 years, and how did we become this species that, traveled the entire surface of the globe and colonized all these locations. So I went to the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. I was in the Department of Evolutionary Genetics, which is perhaps most famous for being headed by Svante Pääbo, who is considered the father of ancient DNA. I was not in his research group. I was in the molecular anthropology research group. So I studied human human population history, modern human population history. And I was in a molecular anthropology graduate program before I heard the term molecular anthropology. And once I heard those two words together, it was this light bulb moment of, oh yes, okay, this is what I'm interested in. This is what I really wanted to know about. But you I guess I kind of stumbled into being something that I couldn't see, but it was really just by chance. And I felt like a lot of pieces fell together after that. So I was focused predominantly on populations in Oceania to try and tease out their population history of migration into near Oceania and then further exploration into remote Oceania, which is thought to be two very distinct migratory waves. So the first one would have happened somewhere around 50 to 45,000 years ago. And the more recent one, part of the Austronesian expansion, more like 3,000 years ago. The degree system in Germany is pretty different from Canada. So I technically don't have a PhD as a doctor of philosophy. What I have is a doctor rerum naturalium, which is considered equivalent to a PhD. There are, I think, about 80 Max Planck Institutes, most of which are in Germany, but there are some spread in other countries as well. None of them are degree granting institutions. So while I did all my work, did all my studies at the MPI AVA, it couldn't grant me my degree. So everyone who studies at one of these institutes has to be affiliated with another university somewhere. My affiliated university was the University of Leipzig. So I went to University of Leipzig, I believe, 3 times, once to register, once to hand in my thesis, and once to defend it. But it's the name on the piece of paper. So I can't even begin to describe to you how much I enjoyed those 4 years at MPI. It was such an intellectually rich environment. I've never experienced anything like that. I think part of that was the time that I was there. Sequencing technology was changing. The Neanderthal genome was in development. They had discovered the genetic signature of the Denisovans, but they didn't even have the name Denisovans yet. They were still calling it the X-woman. It was just a really, really dynamic time in a department and an institute that felt like it was doing really cutting edge research. And I loved it so much. I got a little bit frustrated with my own work. I was still working with mitochondrial DNA predominantly, a little bit looking at whole genome SNPs towards the end, and felt like being a uniparental marker, it wasn't giving me the resolution that I wanted. So it was fine for sort of distinguishing the 45,000-year-old migration from the 3,000-year-old migration. But if I wanted to look at more nuanced details, like what happened between 3 and 2,000 years ago in a particular cluster of islands in remote Oceania, it couldn't really resolve that. And around about that time, I was introduced to the idea of Helicobacter pylori, which is a bacterium that lives in, I think, something like 90% of the world's stomachs. And its genetic diversity mirrors human genetic diversity. Essentially, we have carried it in our stomachs as we have moved around the globe. And it's really an incredible story of coevolution, I guess. So I decided that I wanted to study human history, but not through humans. I wanted to use a different organism, ideally something that had, a little bit more of a rapid mutation rate. And also, maybe less of the ethics concerns of studying human genetics as well when you're looking at microbes. So I wound up at McMaster for a postdoc. So that was a postdoc with the Hendrik Pointer's group, the McMaster Ancient DNA Center. I was based out of the Department of Anthropology, but I interacted quite a bit with the biology department and also the IIDR. And I was there for about 5 years, a little bit over 5 years. So I was a postdoc for all of those years. I was a Banting Fellow for 2 of those years. I was a sessional lecturer for, I think, 4 of those years. I, assisted in helping students or co-supervising probably about a dozen students that went through that lab in those years. And I really, really enjoyed it. I thought that I was going there to study Yersinia pestis and Black Death. I didn't ultimately wind up working on that project, but I think I got my fingers into something like 40 projects over the course of the years that I was there. Some of the major ones that I was involved with were still some human population stuff, mostly because of the experience that I had from my PhD and being able to train students who were looking at those population markers in their study area. And but most of my postdoc, at least the projects that I was driving myself and that I was most invested in, were related to orthopox viruses. So particularly variola virus, which was the etiological agent of smallpox, and then vaccinia, which is the virus that we use to vaccinate against smallpox. And we found those, we found the variola virus through a surprise. We were looking at a 17th century mummified sample from Lithuania that we thought might have TB. And instead we found variola, which was exciting and really shaped the research that I have done since 2016. And then also looking at the viruses that would have been used particularly in the 18th century and 19th century to vaccinate against smallpox. So with those 2 projects, what we've been looking at is essentially the history of these viruses that had major implications on human health and disease. So we think at one point smallpox killed upwards of 25% of all children born. In the 20th century alone, even though we eradicated smallpox by 1978, and it was really not a factor in at least the first world from about 1920, 1930 onwards, we still have more deaths from smallpox than from all 20th century armed conflicts combined. So even though we think of this as not having been an illness that affected us, it still had a massive impact on populations in the very recent past. And smallpox remains the only human disease eradicated by vaccination. So vaccinia played a huge role in that as well. I spent most of my postdoc years on the job market, as pretty much every postdoc does. From 2016 to 2020, well, really 2019, I guess the applications would have gone in 2019. I applied to about 30 positions. Those would have been in anthropology and biology departments. So I felt as though I bridged both of those faculties in terms of what my area of study was, also my experience and my interests. I did come in with some pretty specific needs because of that ancient DNA focus, right? So you need clean rooms, or at least access to clean rooms, which are not cheap to build or maintain or retrofit. So I found that there was a little bit of a pattern in terms of the offers for interviews that I was getting. For the most part, they were anthropology departments. I think that anthropology departments were the ones who saw my research as adding something to what they were interested in or studying, or that might offer their students something new. I think biology departments, unless they were specifically looking for somebody to do ancient DNA, saw me as somebody who wasn't doing kind of basic or foundational research, and on top of that had some pretty big needs in terms of space and equipment. So I interviewed for 6 positions. I interviewed for the very first position that I applied for, which was a surprise, but it was a very specific job posting that they were looking for an ancient DNA person. It was also a European interview. I don't know if you guys have talked about those and how they're different from North American ones. No, they're wild. Everybody who is interviewing goes at the same time. You all sit through each other's job talks. There were 4 people invited to interview for that position. I had previously worked with 2 of them, which meant that it was weirdly congenial and slightly competitive at the same time. I consider it to be one of the best talks I have ever given. And that was because a combination of incredible nerves and jet lag meant that I spent all night running through slides in my head and thinking about slide transitions. And I felt like I nailed absolutely every syllable of that presentation. I bombed the rest of the interview because I had no idea what I was doing. I was not ready to be on the job market. I had a really good presentation, but absolutely no idea what I would do as a research plan or how I would get funding, especially not in Switzerland, or how many students that I would want. But ultimately, it was a really good experience. I was glad that my first experience was so low stakes in a way, in that I knew I wasn't ready to move on to that step, but was also specific enough that I felt like I wasn't wasting my time or anybody else's time. Unlike another European interview that I got invited to a couple of years later that I thought, I don't understand why they've asked me to be here. That was an 8-person interview. There were 8 candidates there. I was absolutely the Monty Python-esque, now for something completely different. They did not have facilities to accommodate what I needed to do. Yeah, I'm still confused about that one. And through those 6 interviews, I wound up with 1 job offer. And I'll come back to that a little bit. It was possibly the right offer at the wrong time. But again, I'll come back to it. And I applied to 1 position with the Public Health Agency of Canada. I had been following their job ads for a few years and hadn't really seen anything that looked interesting or applicable to me. And then this ad popped up and it pretty much listed all of my favorite details about postdocing, about getting to do research, about getting to teach other people how to do science, how to do research, about getting to mentor, being focused on phylogenetics, pathogen evolution, but with the benefits and a salary that I felt like was reflective of my years of experience in education after a lot of years of short-term postdoc contracts and not feeling like I really had very much job security. Okay. So in terms of hiring, anybody who has been hired by the government at any point will probably tell you that it is long and confusing and painful at several points. I'll walk you through how my process went. Since I joined the government, I've been involved in the hiring of probably about 30 other people. Some of these themes are similar across all of them. Some of them are different. So this job that I saw that matched all of my favorite parts of postdocing, but getting paid, was posted in the fall of 2018. I think it had about a 30-day window to apply for it. And the application deadline was the end of November. And it was listed as being a supervisor biologist. It was very clearly an ad that was for a specific position, not one that might be for a group of positions. So there were some very specific questions in the original application. So I submitted that original application through the online portal. And then over the next couple of months, I went through several additional screening tiers. So the first one was, somebody had clearly gone through the applications online and said, like, yes, this person is qualified, this person meets whatever the basic needs are. Then I was told that I was going to be sent a take-home exam. So they give you a bit of advance notice for that. Typically it's about a week, but I think the minimum that they have to give you is 72 hours to complete it. The one that I got was 11 long answer questions. I dug it up the other night to check. And they go through a lot of the basic qualities that are required for hiring within the public service to begin with, and then some that are specific to the particular position. So they will typically tell you whether or not you're being assessed on, something like knowledge or competency or education in a various question. And then for some of those, you're required to have a validator. So if I talked about, here are how I know about assembling viral genomes and talk about all of the steps, then it will say you have to provide the information for someone who can validate this answer. So somebody who can say, you know, you didn't pull these answers off the internet, that this is actually reflective of what you did. And then after the take-home exam, there was an invitation for a video interview. So that was with a 3-member panel. And they told me to block off 2 hours for the interview. My memory is that it was more like 75 minutes, maybe 90 minutes. But it was a long interview. It was a long period of time. They sent me some of the questions in advance, and actually encouraged me to have notes going into it so that I could really make sure that I addressed some of them. But other questions were really about how did I answer them in the moment. In May of 2019, so essentially 6 or 7 months after I submitted the original application, I was informed that I was considered the right fit candidate. Now, it's important to know when you're being hired by the government that being told you're the right fit candidate does not equate a job offer. What it means is they want to hire you. They cannot yet offer you a job. You have to clear security and possibly medical before that. Security is a nightmare. Like every hiring manager will tell you, security is the worst part of hiring because you don't know what's happening. You send off forms and then it's essentially out of your control. And it's really a part of the system where it's clear that the branches don't talk to each other and there's no way of tracking it. It's not like being able to log into Mosaic and see who's checked off on an expense report. It just kind of goes out into the ether. And you can pester people for updates, but there's no way to really know what's happening. There were some complicating factors to my security clearance. So the first was because I was being hired to work for the NML based out of Winnipeg, I was required to have secret clearance. If you're based out of the NML at Guelph, you're only required to have reliability. I had also lived outside of Canada within the last 10 years. So I'd been in Germany. Which means I had to get a police report from Germany. I had to give them information about my German landlords and all of these contacts. And I don't know what they did with that, but it slows things down when you live outside the country. I had to provide personal references. My former employers were contacted. In terms of my education, I had to provide a translation, like an official signed, validated translation of my German diploma. I had to pay a center at the University of Toronto to do an assessment that essentially said that my degree from the University of Leipzig was equivalent to a PhD granted in Canada. As I mentioned, there was the criminal background checks, both in Canada and in Germany. And I had to do an interview with CSIS that they don't warn you about. They just called me one morning, 8 a.m. I hadn't had any coffee yet, and grilled me for about 45 minutes on a variety of topics, including what were all of the countries I had visited in the last 10 years, and I wasn't allowed to look at my passport or a map for it. It wasn't fun, but it's part of it. I had to get medical clearance because my position at that time involved being near labs. It was a lot. I'm sure there's actually details about it that I've blocked entirely, but it was a long, confusing period where nobody knew what was going on. At the end of 2019, I decided to leave my postdoc. So my contract was ending I kind of felt like I was expecting to get this job, and because I was expecting to get this job, I wasn't taking on extra projects. And so I felt like things were kind of getting a bit stagnant in terms of research that way. I also felt like I hadn't had a vacation in 10 years and wanted to take a vacation. In hindsight, it was the worst possible time. I managed, one leg of all of the vacations that I had planned. And I had also applied to this position in the fall of 2019 because, again, it was something that was super specific to what I was interested in and my skill set. And I interviewed for that in February of 2020. And in March, they offered me that position. So it's, as you mentioned, I think it was the right position at the wrong time. So I, to this day, think that that department was possibly the best possible department for me in an academic sense. But I was feeling pretty worn down from the academic experience in general. Also the government security process, and felt like I'd invested a lot of time into that. In hindsight, this next part seems really ridiculous, but that department seemed wonderful, but the town it was in seemed small. And, at that point I was, I think, officially entering my mid-30s, and I just felt like everybody was telling me that it was a wonderful place to be there with, small children, young families. And I thought, but that's not me. I felt like life was going to be really small if I moved there. And like 2 weeks later, we all got locked in our houses for, I can't even remember how many months, making it seem small seem so ridiculous in hindsight. So I essentially pushed the NML to tell me what was going on. I said to them, I have this other job offer that's an actual job offer. It's not something saying that I'm the right candidate, but all of these other things need to happen before I can be hired. They're saying, we want to hire you. And that kind of pushed the NML to really ask a lot of people some questions. And it turned out my security had been cleared 2 months earlier, but that piece of paper hadn't made it to one desk who hadn't made it to another desk. And so nobody knew that I was security cleared. So by the end of March 2020, I was security cleared. We're, 18 months after I submitted my original application now. And again, we were so naive in March of 2020 that the official line that they gave to me after I was considered, security cleared and all of the processes were going forward for my hiring was sit tight and wait for this to blow over. A couple of weeks later, I got a different message that said, we need you to get here now. So while everybody else was, bleaching down groceries and leaving their apartment once every 2 weeks, I had to try and figure out how to rent an apartment in a city that I'd never been to, how to get to that city in a pandemic. And yeah, it was a stressful couple of weeks. So in May of 2020, I rented a car, packed up as much belongings as I could, and drove for 3 days to Winnipeg, where I then spent my first 2 weeks in a new city, quarantined in a rental apartment. And when that was done, started going to work at a job that, even though I'd been waiting, 18 months and had thought that it was my job essentially for a year at this point, no longer really existed in the way that it was described to me, because all of the operations at the NML had become COVID focused at that point. So there was nothing happening but COVID. And my job had really been to perform support for all of these other groups. So it was a strange time. That position that I started, which, if you remember from the original ad, was called supervisor biologist, was more formally called the head of genomics. That was the specific job title. And that's still considered my substantive position. So that's the permanent job that I hold with the government, even if I haven't specifically been in that job for more than 2 years at this point. It was an unbelievable amount of meetings. and all sorts of new acronyms, most of which were related to CanCoGen, which you may have heard about, which was the Canadian COVID Genomics program. So the federal government essentially gave PHAC and the NML a lot of money to try and figure out how to monitor COVID within Canada. And so during those months, we went from the original flavor of COVID through the alpha and the beta and the gamma variants. There was the mink outbreak, which caused a little bit of panic at the while. But it was a strange position to be in because I felt like I had a lot of skills. I was being helpful where I could be helpful. But I was in a job that didn't really have anything to do at that moment. And I think part of that was that nobody really wanted to put too much pressure on me as the new person, and nobody really wanted to actually claim me for a new position because they knew I had been waiting so long to get that job. They didn't want to try and change what my job was. I think when I think about that transitionary time, it was a little bit like Alice in Wonderland. The caveat for all of this, of course, is that it was the early days of the pandemic and it was a weird time. No matter which way we think about it, it was just weird. So I can't say how much of this transitionary experience was about me, about that position, and how much of it was just about where the NML was at that time during the pandemic. But it was very much like learning a new language. I felt like I never understood what was happening. I didn't understand the words. I think maybe it would be like jumping into an advanced molecular biology class if you've never taken anything before and hearing people throw around PCR, DNA, fish, etc., and not knowing what any of that means. A lot of frustration, partially because I felt like I had skills that weren't being used. And also, things just take a different time. And it's interesting to watch new people come in now and have those same frustrations and realize that I've become completely zen about them, that it's something that needs to happen. It's a process that needs to happen. It'll work out. It just might not happen immediately. That, there's not that immediate gratification with a lot of things. Fundamentally, the function of the public service is different. I know it's built right in there. It's about service. So all of the work that we do has to somehow connect to the mandate of either the group that we're in within the NML, to the NML itself, to the Public Health Agency of Canada, or maybe to Health Canada, but that divide gets a little bit murky sometimes. And it was really a new way of experiencing the healthcare system. So feeling as though we were, all in this project together about tracking variants across the country, tracking COVID across the country. But of course, healthcare is a provincial responsibility in Canada. So as the federal government, we couldn't actually ask directly for that data. It wasn't an assumption that we could have it. Everything has to go through each individual province and has to be approved. So there's a lot of legal agreements. And that varies from province to province. In some cases, it can be signed off by the head of the provincial public health lab. Maybe it has to go to the chief medical officer. Maybe it has to go to the minister of health. It varies from place to place, province to province, and then the territories are different again. So after essentially a lifetime of feeling like the Canadian healthcare system made a lot of sense compared to alternatives, and that it was universal and we could access it all the time, all of a sudden it felt like there were all of these barriers to getting the information that we needed to be able to track and manage the pandemic. In March of 2021, I switched to a new position within the NML. So this is what they call an acting assignment. As I mentioned, my substantive was and still is as head of genomics. And this was a temporary assignment that I took for about 2 years as a science project coordinator with the CCGP, which stands for the CPHLN, which is the Canadian Public Health Laboratory Network. So that first C stands for 5 other letters, which stand for 5 other words. COVID Genomics Program. And what we did with that project was essentially build a genomic surveillance system across the country. So we hired staff and embedded them within provincial public health labs to get the provincial public health labs sequencing their own COVID genomes. There were a few labs that were doing that at the time, but we essentially provided the human resources and the technical resources, and in many cases, reagents to be able to facilitate that. It was a lot of staffing. Like I said, I think I hired about 30 people or participated in the hiring of about 30 people, which is interesting. Also, we did that really fast. We hired 30 people in about a year, which in terms of the government is really, really fast. We hired some of them within like 2 or 3 months, which is like practically the blink of an eye in the government. And a lot of that role was about problem solving, problem solving from a remote position. So not only were most of us working remotely, but we were supervising people who are in different provinces. So, maybe talking to a province out west or talking to a province on the East Coast about what their problems are and trying to figure out how we can, either change their library prep protocols or figure out their sequencing chemistry or their data analysis so that everything is up to a standard that is continuous across the country. Last spring, Mpox became a thing. I thought that studying variola virus was just this kind of strange thing that I enjoyed doing that nobody would really care about. And all of a sudden it became really important. So last spring, I had the genomics lead position on the Mpox surveillance in Canada. So all the Mpox samples that were sent for testing at the NML, if they were selected for sequencing, that data ultimately came through me for analysis, interpretation, reporting to senior management, and ultimately releasing to public repositories. Esoteric became really important, which still seems so crazy sometimes. Okay, so my third position at the NML. I've been with the government for 3 years now. This is essentially my third position. It's again an acting assignment, so it's not my permanent job. It's something that I'm holding for a particular period of time. And right now I'm the acting chief of COG. So COG stands for Computational and Operational Genomics. So it's a maternity leave cover essentially, but it's a maternity leave cover at an interesting time. So COG up until early last month was the computational biology unit, and we were located within the bioinformatics section. We're now essentially going through this fission where we've been promoted from a unit to a section. So where we used to be sort of underneath the umbrella of bioinformatics, we're now like a sister group. So there's a lot of transition stuff that I'm managing for that. And it's been a really interesting experience. I feel as though I'm getting to learn all of these new skills in terms of, for the most part, really financial management and reporting within a government structure, which doesn't sound like it should be exciting, but it's strangely interesting to understand how these things work sometimes. And COG is fundamentally an enabling program. So we're not a research program, we assist other programs. So we do a lot of routine work. Still, most of that is associated with COVID. So we make sure that the data that comes in is processed appropriately, that the metadata is curated, that it's uploaded to national databases, public-facing databases. We'll do ad hoc analyses if they are requested by surveillance teams or animal management, or because Teresa Tam wants to know what's going on with wastewater that week. We do a lot of pipeline development for various groups. And fundamentally, we're about supporting other groups within the NML, occasionally within the provinces, but mostly within the NML, either as they adopt whole genome sequencing or as they move into a new pathogen. So as an example, we're trying to help wastewater surveillance scale up and automate its processes. so that it can really broaden its outreach and efficiency across the country. Influenza monitoring has typically been based on PCR typing, and we're assisting the group associated with that to really move into whole genome sequencing and scale up their operations so that it looks something more like COVID surveillance. Or, new pathogens like Mpox last year. So while I was doing that genomic leadership position still through the CCGP, I was interacting a lot with the computational biology members at that time. Or, also last year, a request to make sure that we had appropriate pipelines that existed, were operational, and were validated in case polio cases appeared in Canada after there was a few that were identified in New York and the UK. So making sure that we were ready in case that became a national concern. In addition to my role with the NML, I maintain an adjunct affiliation with McMaster. So I've had that essentially since I began my role at the NML. So with that adjunct affiliation, I continue to support grad students for the McMaster Ancient DNA Center. Typically, those are grad students that were around in my days as a postdoc, but some of them are also newer students. With that adjunct affiliation, I can continue to apply for and hold Tri-Council grants. So I've got an insight development grant that just finished and an insight grant that I'm working on, which is all still, again, related to orthopox viruses. And it gives me essentially permission through my job with the government to continue to be available for seminars, guest lectures, things like this. That said, I have to declare that through the Conflict of Interest Office with Health Canada and FAC, which essentially means that they have to feel confident that neither one of my positions has any really financial influence over the other, and it's not a concern at all. So then I thought I might very quickly, I guess, walk people through how you might look for or apply for a government position. So you want to start at jobs.dc.ca, and there'll be a little bar that says find government jobs. I like to sign up for the email alerts just to know what's out there and to make sure that it goes directly to my inbox so I don't have to check the website regularly. You can filter it for a particular agency like CFIA, Health Canada, or PHAC, or you can get everything and just understand what the breadth of the public service is. There's no other way to know that occasionally prisons in BC are looking to hire tailors, but this is how I know that that exists. I would say don't be afraid to apply early, go lay down. As I said, the process is really slow. And if you get through a partial application, that can still be reused. So it's worthwhile applying for anything that you think might be interesting, even if you don't think the application deadline might align with your graduation date. When you find an ad, there'll be a header section like this. It'll have information about what the job classification is, and it might have a job title or it might just reflect the classification. And there should be a job description as well. So here it says that it's a biologist 3, then it also tells you that it's a bi 3. So essentially it's saying we're looking for biologists. The classification here is 3. But you might also see something that is listed as the class bi 3, but they give supervisor biologist as the title, or research biologist or lab biologist, something like that. There's 2 different types of ads. You can either get an inventory, which isn't specific to a specific position, but it's rather about creating a pool of qualified candidates that multiple positions can be hired from, and managers can essentially say, please give me all of the applicants who are in this pool. Or there might be a hiring process that's specific to a particular opportunity. And in that case, the questions are usually both more detailed, but also fewer in number, because it's really about finding one person for one job. So in this case here, we can see that this is a bi 3 inventory. The fact that it's giving the location as various is also an indication that it's not for a single position, but rather they're looking for a pool. They'll also tell you your salary range. And if you combine that with the job classification, like the bi 3, you can go to the Treasury Board website and look at what the pay table is and have all the information. So it'll give you this range because the pay table will have multiple steps in it. All of the jobs will try and start you at step 1. Nobody will tell you that you can ask for a different step, but you can ask for a different step. You just have to justify it. They won't necessarily approve it, but you have to ask. They won't ask you if you want to ask, if that makes sense. There'll be some description underneath that. There'll be a lot of description underneath that, to be honest. I'm just kind of picking a couple of options here. So here that you can see that, again, it's really telling us that they're looking for multiple opportunities. This is an inventory, not a specific application. Your application may be accessed at any point. So even if the deadline for the application says, you know, July 13th or whatever it was on the following slide, they might start pulling applicants from that before that. If a manager says, I'm really looking for a by 3, I need them right now. And then here are the various kind of specific job titles that might fall in there. And here again, it's saying potentially access as early as May 5. There will be a whole bunch of information underneath it about various criteria that are considered. Some will be essential, meaning you have to have it to be considered for that position, and others will be called asset. So an asset is a nice to have, not a need to have. And it might be something that they use to filter down the number of applicants if there are a lot, but it doesn't mean that not having that asset means you won't get the position. So, you know, if I'm looking for a biologist and I have 3 essential criteria that I list and 2 assets, and nobody has any of the assets or only 2 people meet the assets, I'm not going to ignore people who don't have them. Alternatively, if I look at it and there are, you know, 100 people who meet the assets, the essentials, but there are 10 that have the assets, I might only pull the asset applications and not look at the others as well. When you're applying to these, it feels like a lot of text. I know that it's a lot of text, but it's really important to read them. And there's some tips on how to apply. So if you can try and echo the language that's in the statements, that's really important because people will key in on particular words. You really want to pay attention to whether a list uses and or, because that changes whether or not, you know, you have to have all of these qualifications or you just really have to have one of them. If you have to have all of them, you need to address all of them. Otherwise, your application won't be considered. It's really great to use I statements and action verbs. And most importantly, you really, really have to be specific. The other thing to know about the government is that they don't consider education as proof of qualification, that you really need to tell them why this is. You can't just say, I have a degree in molecular biology, ergo, I am a molecular biologist. I'm equivalent, I'm qualified to be a molecular biologist. You need to explain what your experience is. You want to try and avoid acronyms. Don't refer to previous answers or your resume because they might all be considered separately. Try to be accurate. You know, don't exaggerate. That's something that for particular people will really, really irk them a lot. Same thing goes for proofreading. Again, your application might not be read from top to bottom by one person. Different people might be reading different parts of it. So proofread it and make sure that you're really answering every question individually, even if it does sometimes feel repetitive. Yes, okay, again, pay attention to and versus or. Okay, essential knowledge means you have to know things. This one about the mission and mandate of the Public Health Agency, I have sat in so many interviews where people could not answer this question. If you are applying for one of these jobs, I strongly suggest that like 5 minutes before your interview, you Google the Public Health Agency website and look at the bullet points for what it says that the mission and mandate and objectives are, because somebody will ask you, and if you have a great answer, it will get you brownie points. When you click in to actually look at the application, there'll be all of these different parts that you have to fill out. It is lengthy. you should expect to spend a couple of hours on this. It's not something that you do right before or in a rush. Like, you need to dedicate time for it. Like, I won't lie, it's not fun, but you got to do it. There will probably be a subset of first questions where they do expect long answers. So recent and significant is something that they will frequently use to talk about experience, and then they will give you a definition for what they consider to be recent and significant. They will want specificity, so concrete examples. If the question is, do you have experience analyzing and assembling bacterial genomes, don't say, yes, I have assembled and analyzed 100 Pseudomonas genomes. Talk about what the data was, how it was generated, what programs did you use to assemble it, how did you call your consensus, what quality checks did you do, how did you validate them. They really give them all the details, because I have seen so many applications from people that I know are really smart and could do these tasks, but they answer the questions poorly, and because of that, their applications don't move forward. Yes, and again, breadth and depth is important. There will then be a whole lot of these screening questions, and I'm not joking when I say there might be 100 of them. And these will just be yes or no, but these are the questions that people might ask for more details on at a later date when they're looking for a particular position. So in these ones, don't be afraid to say no, especially if they're a really technical specific one. So, if you're a molecular biologist who does phylogenetics of viruses, and there's a question about, have you ever done fluorescent in situ hybridization? Don't be afraid to say no. It doesn't mean your application will die. It just means you likely won't be screened in for any jobs where fish is important, and you don't have that experience. So you probably don't want to be anyways. But these are questions that if you answer yes to, you will likely be asked for more details at a later date. So when you have long answer questions, the government loves the STAR method. Sometimes they will explicitly tell you to think about the STAR method, but if they don't and you use it, again, people will love it. So situation, task, action, result. That's your little mnemonic, just in terms of how to structure a response. And that will really kind of fit or conform to their expectations.
We have a question in the chat.
Yeah.
You presented a lot of information about job posting. Do you have any insight on postdoc positions within the federal government or PHAC? I have yet to see any postdoc positions for Government Canada job on Government Canada jobs. So on the website, are they on a separate page?
So they won't be on a separate page. They're not common. A postdoc position typically has to be funded through a specific grant. So it's again, you're not really staff. There's a research affiliate program, which I believe had a posting go up a couple of days ago, and I think it includes postdocs. It might just be grad students. But in general, I would say postdoc positions, with at least within my experience within the NML, are not terribly common. So this is just sort of a roadmap of what the steps are to kind of getting to that appointment. And again, maybe this happens in 2 or 3 months, maybe it takes 2 years, but there's a whole bunch of stages that happen. And when it feels like there's nothing being said for a long time, it's usually because there are wheels spinning in the background. I will skip this. I thought I would give a couple of observations of what I've noticed in the last couple of years as the NML has grown tremendously, largely in response to the pandemic. There's been a lot of hiring and a lot of new sections that have been formed in many respects, because there's either this new problem that we need to solve, like wastewater, or because groups have gotten so big that they need to split out, kind of like COG from bioinformatics. And there's a few things that I've noticed in terms of who the people are that are kind of selected to have those new positions that are really effectively promotions. So unscientific, anecdotes, my own observations, huge caveat again. With exceptions of very, very high ranking positions like executive directors, VPs, etc., they're internal staff who are picked to kind of get the tap and move upwards. These are people who are reliable. They are showing up. They are there when they're supposed to be. They're on time. Their work is completed on time. They're people who, when presented with a problem, don't say, here are all the faults, here's why it won't work. They're people who say, okay, let's figure out how we can come up with a solution, who can try and figure out how to make things work instead of just kind of having this mental block and saying, it can't be done. They're people who work well with others. That might sound self-evident, but I think a lot of people expect that it's the person who has the most publications or the person who yells the loudest in a meeting who kind of gets moved up the chain of command. And in my experience, that isn't true. Instead, it's people who are excellent communicators. And I mean that in terms of all forms of communication, not just, written or oral. It's about presentation and body language, and about listening to others and being able to digest information and translate it or communicate it back to people as necessary. And I think really what that says is that these people who are kind of chosen for these new tasks, for making new projects, making new groups, because they're thought to be good leaders, it's because people recognize that they're already good team players. That there are people that will kind of get moved into this new position. Okay, this is the end. So 3 years. I don't think I mentioned this. I was not super happy to originally be told that I was getting a job at the NML. It took me a long time to reframe being offered a job as something other than failure to get the job that I thought I wanted and that I had been training for my entire adult life. And I wasn't always happy after I joined the NML, particularly in the early days when things felt really chaotic. But 3 years later, I can very, very confidently say I'm happy with the decision I made to take this job. I have a work-life balance for what feels like the first time. I never feel guilty about turning off my workstation at the end of the day or on the weekend or taking a vacation, compared to, grad school or postdocing, when I take a vacation or a vacation, a long weekend. And by Sunday, I was already thinking about work. And on Monday, I was feeling really, 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 really guilty about not being at work. I think that the other benefit is that my employer has been very flexible. I moved to Winnipeg and then I left Winnipeg. It wasn't making me happy. So I'm now doing a job that I like, that I think I'm pretty good at. And it's let me move back to Newfoundland, which is a place that I love, which again feeds back into that work-life balance. But I never thought that I'd get to come home until I retired. And so, yeah, being able to be here, doing a job that I like, that I think I'm good at, feels like I'm really valued and that they trust me to do that work. And I'm very grateful for it. I do sometimes really miss academia and research, but I think that the adjunct keeps me connected to that enough that I don't need, maybe the federal position doesn't give me the endorphins of, a scientific discovery and that excitement with a new result, but it also doesn't give me the cortisol of feeling like I'm not doing enough and that I'm always behind on things and I need to be working more. So I think I have the balance that I need and I like, and I really thought that I was going to be short. I'm so sorry that I've gone over time.
That's okay. We really enjoyed it, and you had tons of valuable information for everybody. We're already a little bit over time, but there's one last question in the chat. So,
okay.
So once you got your job, Are there opportunities for more training after you start?
Absolutely. So I think that might be the slide that I skipped. One, there's a lot of mandatory training. You're going to have to do all of these courses about who we work for, about how to access your pay slip, about respect in the workplace. You know, for the most part, they have a lot of good content. There's then additional positions that are only advertised internally. Most are advertised externally, but there are some, and that really is where a lot of acting assignments or secondments come from. Most of your positions will have a learning plan associated with them. So that typically would include funding for attending a conference or maybe an additional training course. There's opportunities to keep learning that way. One of the things that I found really surprising when I started is people kept telling me like, oh, I'm in this group now, but I used to be in that group. And before that, I was in this group. And before that, I was in this group. And I thought this was a sign that people hated their jobs and that all of these groups must be dysfunctional if people were moving between them all the time. Like, you know, after years in the academic system, isn't the goal to get the job and then stay there until you die? And instead, it's really about people kind of saying like, hey, I've been doing this for a while. I've had fun. I want to learn something new. I want to experience something new. I want to, you know, try something new. So there will be lots of kind of moving between and sort of internal rearrangements. Yeah, so there's opportunities, I would say, both internal in terms of changing positions and then also, you know, conferences or training. Yeah.
Awesome. Okay, so we're out of time, and I'm just going to thank Anna Duggan for an awesome talk that covered how to apply for jobs, where to find jobs, how to make yourself stand out, all things that are very valuable to everyone. You're getting a virtual clap here.
Thank you.
Thank you so much for joining us, Anna.
No problem. My pleasure.